better.
Somebody was asking me earlier that if the exam will be done today, but not today, so maybe Thursday or next week. Yeah, and I was wondering if you had any question on intracellular signaling last week. No. Okay, so let's move on. Today's topic is early brain development. So basically, we're going to learn how to make this. And it's from chapter 22 of your textbook. So it's obviously very complex. So how to make this complex structure is what you're going to learn today. So as you can imagine, actually, let me start with asking you a question. How many neurons are there in the human brain? How many of you think it's more than 1 million? Yeah. So other people think less than 1 million? Yeah. So yes, the answer is it's over 100 billion neurons. So there are so many of them in your brain. Are you using all of them? So obviously, as you can imagine, there are so many different types of neurons in our brain. So the question is, how to generate so many different types of neurons? And this question can be raised to which genes to turn on. And we are going to hear a lot about transcription factors. So what is a transcription factor? Transcription factor is a protein which goes to nucleus and recognizes certain DNA sequence, binds to it, and then initiate transcription. So what is a transcription? Transcription means synthesizing messenger RNA using a complemental DNA as a template. And then this messenger RNA will be translated to what? Proteins or peptides. So that is called gene expression. And transcription will be mediated by enzyme called RNA polymerase. So let's think about the transcription. So the portion of DNA which will be used to make messenger RNA is called gene. And in front of the gene, there's a region called promoter in the DNA. And polymerase, RNA polymerase will bind to it and then initiate transcription. But RNA polymerase doesn't always bind to it and then initiate transcription. It requires a cue. And that cue is provided by transcription factor. So transcription factors will bind to a certain DNA sequence inside the promoter region and then start forming a protein complex as shown here, which includes proteins called activators. Activators will bind to another DNA sequence called enhancer. Enhancer is usually located further upstream of polymerase. So as a result, DNA will form hairpin loop. So this hairpin loop structure, together with the right kind of protein complex, will be recognized by RNA polymerase. And that's how you start transcription. So what kind of neurons to make means basically which sets of genes will be transcribed and then later translated. And there are two important things to determine which neurons to make. One is position. So where in the body determines what kind of neurons to make. And another is timing. So at what stage of the development determines what kind of neurons to make. And the nervous system develops as the other parts of the body develop. So we have to know a little bit about embryology. So let's think about embryology. So as I mentioned, brain has a lot of different kinds of neurons, but our body has a whole lot of different cell types. But if it's your cell, they all have the same set of genes, your gene, right? So it doesn't matter if it's your cell, a hair cell, skin cell, or gut cell. They all have your gene, same set of genes. Just the difference is which sets of genes are expressed, and that determines the cell type. And we all started out as one cell, and that one cell divided and divided, and eventually made all the cell type in our body. Have you ever heard of stem cell? If you pay attention to newspaper science section, you will often encounter the word stem cell. So let's talk about stem cell a little bit. So when they say stem cell, they often refer to embryonic stem cell. So two criteria has to be fulfilled to say stem cell. One is the ability to replicate itself, which is called self-renewal. Another is the ability to make all the body parts, which is called pluripotent. So like I said, we all started out as a one cell. So we all used to be, and then that one cell made all the body parts. So we all used to be stem cell. Then what's the big deal? Well, the big deal is scientists found a way to keep them in the culture dish. And that's kind of useful because then if you want, you can give them cue and then make certain cell type. Why is it useful? Well, let's think about Parkinson's disease. Parkinson's disease is the most common neurodegenerative disease in humans, which is caused by a loss of very particular neuronal pathway. Dopaminergic neuron in the midbrain, the region called substantia nigra, is particularly lost in the disease. So if you can make a midbrain dopaminergic neuron from stem cell and then transplant it, and then you might be able to cure the disease, right? Well, in fact, in the animal model, transplant stem cell derived dopaminergic neuron can really rescue the symptom of Parkinson's disease. Yes? Yes, you.
How would that, wouldn't it be really hard to transplant it because of like the different synapses that are also specialized?
That's a very good question. So she asked, how can you transplant? Wouldn't that be difficult because it has to make a right connection? Yes, that's exactly right. Well, the trick is midbrain dopaminergic neuron project far away, and the target cell is in the striatum. So making it exactly like that is very difficult, but if you transplant the dopaminergic neuron into striatum, it doesn't migrate, it doesn't have a right cue, it doesn't migrate well, but it can make a connection with the right target right there. So that's how it works. Yes. So yeah, so that can be useful. So it's a powerful clinical potential. So that's why they are excited about it. However, there is a problem. If you transplant somebody else's cell into your body will recognize and attack. So the patient has to be immune suppressed. But if you are immune suppressed, you catch a cold, and then that could be life-threatening. You have an immune system for good reason. So ideally, if you can make your cell and then transplant it, that's perfect, right? Well, this person, Shinyu Yamagata, found a way to do so. So he took differentiated cells, such as a skin cell, and then forced them to express several genes, and then this cell will turn into embryonic stem cell. So we call it induced pluripotent stem cell, iPS cell. And that's what they are excited about. So he found this method less than 10 years ago, and he already got Nobel Prize. So you can tell what impact it had. Okay. Another thing about I'd like to mention about is this one, neural stem cell. Because we are going to deal with this type of cell a lot in this class, which is also called neuroprogenitor cells. So neural stem cell is like an embryonic stem cell. It has the ability to self-renew, but it doesn't differentiate all the body parts. Instead, it only generates neuronal cell type, which is neuron, astrocyte, oligodendrocyte. So the cell fate will be a little bit more restricted than embryonic stem cell. And one more thing I would like to mention is this. Cancer cell. Cancer cell has an ability to self-renew. In fact, they self-renew like crazy, and that's the problem. And if you look at embryonic stem cell and a cancer cell, they are kind of like, they are kind of similar. The difference is embryonic stem cell could generate different cell type, but cancer cell cannot. So a lot of scientists think origin of cancer cell might be this type of cell, embryonic stem cell. And something went wrong, and they lost the ability to respond to the cue and to generate different cell type. So that was the stem cell topic. And like I said, we all start from one cell, so I'd like to show a video of how we start our life. So this is the one cell that we start from. So you can see the cell division very clearly. So this is really the initial step of our life, and as you can see, there are so many cell division going on. And you might have noticed toward the end of this video, the total size of mass doesn't really change. So they just divide and makes a lot of small cells. So this stage with a lot of small cells is called blastula. And blastula, next step is, will turn into gastrula. Gastrula has different layers. The most outside layer is called ectoderm. And the inside layer, which is highlighted in the green, is called ectoderm and endoderm. And between ectoderm and endoderm, there will be a third kind of cell called mesoderm will be filled. So those three layers are called germ layers. And they will generate different parts of our body. So can you imagine where the nervous system is generated? It's a little bit surprising. It's actually this one, the most outside one. And when you think nervous system is always inside, it's kind of surprising, but you will see how nervous system will be generated from ectoderm. And another thing they produce is, well, basically everything outside, hair, skin. What about endoderm? What kind of body parts they create? Which is basically from mouth to the other end. Basically, yes, stomach, intestine, colon will be made from endoderm. And mesoderm will make muscle, and skeletal, and blood vessel system. So, I would like to show you a video of how human embryo will go through this step. So, going from blastula to gastrula is called gastrulation. So, I'd like to show you the video of gastrulation of humans. So this step happens 2 to 3 weeks of our life. So you can see this blue one is the ectoderm. And from this part, you will see something like a scratch. This is called a primitive streak, and that will extend, right? And beyond that part will become neural plate. And then this will be the brain, and this globe is neural globe. And then next we're going to see how mesoderm will be developed. So they are going to show the cross section. So that's the primitive streak. Epiblast means the part that makes our body. Hypoblast is outside our body, like yolk sac and the placenta. And then the cell will be generated from primitive streak and then fill the space. That's endoderm. And then between the ectoderm and endoderm, third cell type, mesoderm, will generate it and fill the space. It's like a chocolate sandwich cookie. So that's how the 3 germ layers will be formed. And one more important thing it will show, which is the nodal. So this is the primitive streak. At some point, there will be a structure generated and then go underneath. And this will instruct development of neural plate. So it's important. So a lot of things are developing at the same time. 3 layers formed, and then one by one, different things appeared. So this is day 18's view of our body, and as you can see, this is the node code. If you cut section, it looks like this, but it's really a long bar. And that would instruct ectodome to create this one, neural plate. And this portion will start generating a neuron in the end. Now, at this point, as you could see, there is a basic plan of body axis already laid out. So the primitive strip develops this way, and this part will be a head part, which is anterior. And the other side is called posterior. But in the brain, often we think it's the nose part over the back side. So that's rostral and caudal. And also, of course, from middle to the side is medial lateral. And the side that's like back side is dorsal, and then ventral is the belly side. So those terms are very often used in neuroscience, so please get familiar with those. Then what happens is that this neural plate will make a globe. We saw a neural globe, so it becomes like this. And then this part will be fused together and then pinched off. So it will become tube. It is called neural tube. And then this fused part will become, will be around here, will become a neural crest. And if you look at this neural tube, the portion which is close to the surface is called okay. So the close to the surface is called the roof plate, and on the other side is called the floor plate. And those are going to be very important portion because that's like an information center and sending signal. So the dorsal ventral axis will be instructed by this roof plate and lower plate plus nodal. And how do they instruct them? Well, they will send signaling molecule, which is particularly called morphogens. What is a morphogen? Morphogen means it induces cell differentiation. And those cells will be determined cell fate. But not in a random way, organized way. So you can make ordered structure. So, what kind of signaling molecules are they secreting? Let's look at it one by one. First, let's look at this low plate and nodal. They secrete this molecule called sonication. And this picture is a technique called in situ hybridization. So, do you know what you can detect with this technique? this technique detects messenger RNA of certain molecule. So in this case, it's messenger RNA of sonic hedgehog is visualized. So you use probe to bind to this certain messenger RNA sequence, but this probe is conjugated with some kind of enzyme. And this enzyme, if you add substrate, will make some chemical reaction, and as a result, it makes color. So this dark color is done by this enzyme. And as you can see, sonic hedgehog messenger RNA is pretty restricted to flow plate FP and the notable FCP. So, sonic hedgehog is originally found in a Drosophila fruit fly. And the wild type Drosophila larvae looks like this. And then they found if it lacks this protein, and then the larva becomes like this. Doesn't it look like something? Right? So they decided to say, well, this is a hedgehog. And they found there are 3 subtypes of hedgehog. So they decided to name each of them. Well, first 2 of them named after the real hedgehog kind. One is Indian hedgehog, the other one is desert hedgehog. But then they ran out of name for the 3rd one. And they went like, have you ever heard of this computer game? It was very popular back in early 90s, I guess, when they found this molecule. So, 3rd one, sonic hedgehog. So scientists get to name new protein when they discover, and they have a lot of fun naming them. So how does this sonic hedgehog work? So sonic hedgehog has a receptor called patch. When it binds to patch, there's a co-receptor called smoothen. They get together. And then that will send signal inside intercellular signaling. And then as a result, this transcription factor, V1, will go inside and then bind to DNA and then turn on the gene. So you will, as you will see, we're going to learn a lot about transcription factors. And they often have this name with 3 letters and number. So it's pretty intimidating looking, but they are just transcription factors. Please relax. And V1 has a DNA binding motif, which looks like this. So 2 beta sheets is on one side, and then there is an alpha helix, and there are residues which will chelate zinc ion, just like this. So they name it zinc finger. And this DNA binding motif appears very often in different transcription factors. So if you hear zinc finger, then you will know, oh, that's DNA binding motif. So V1 has that. Okay, so that's the ventral part signaling molecule. What about the dorsal part? Dorsal part has another morphogen called bone morphogenic protein, or BMP. BMP has a receptor, and it binds to the receptor, and then that signal will send to inside the cell, and this protein called SMAD get together with this another protein called COSMAD, and then they go to the nucleus, bind DNA, turn on the gene. So, as you can see, there's another protein binding BMP called noggin. What it does is it soaks up the signaling molecule, so you can prevent the receptor activation. And why do we need something like that? Well, let's see where noggin is expressed. Again, this is in situ hybridization, which detects messenger RNA of noggin in this case. And where is it expressed? You can see roof plate, lower plate, and a notochord. Those are the places that sense morphogen, the information center. And why do we need that? Well, as you can imagine, because from the name, bone morphogenic protein, BMP does not just creating different neurons, but very important role for making different cells. It's a very strong morphogen. And you don't want to change the cell fate of secreting morphogen type of cell type. If they change the fate, and then they can't keep secreting the morphogen. So you don't want that. So it makes sense that they shut down the BMP signaling by itself. Okay, so what kind of neurons will be made by this gradient of two morphogens? The spinal cord is the place where this was completely, intensively studied and well understood. So in the dorsal part will become close to the roof plate will become sensory neurons, and then the ventral part will become motor neurons, different type of motor neurons, and the set of transcription factor which determines these cell fate will be this, and it's nicely color coded. And like I said, it's intimidating to see so many 3 letters and a number, and some of them even have decimal, but just transcription factors. And as you can see, some of them are overlapping, like this one, overlapping. This one's overlapping too. A lot of them are overlapping. So the cell fate is not determined by just one transcription factor. It usually requires sets of them. So neural tube creates different neurons, but another place that creates neurons is neural crest. So let's look at neural crest next. So neural crest is here, and this part of structure creates neurons like sympathetic neurons, autonomic, parasympathetic neurons, autonomic neurons, and also adrenergic gland, adrenal gland. So those are going to pretty far away from the central generating part. So it has to migrate. But neural crests are actually here and sticking each other through this molecule called cell-cell adhesion molecule, cadherin. So when they are ready to migrate out, they don't want this cell adhesion, strong cell adhesion. So they sort of want to break the connection to each other. And actually they do. When they're ready to migrate, differentiate, migrate, they turn down the expression of cadherin. How do they do that? Well, they express replacer for cadherin gene, which is called snail. And if you look at again, in situ hybridization, or against snail messenger RNA, you can see neural crest is highly expressed. Now, this protein was discovered also in the Drosophila, and if you like this protein, the Drosophila will looks like this. Do you see this? Actually, I see several of them here and here, maybe here too. So that's why they call it snail. So we've been looking at two morphogens, BMP, sonic hedgehog, and there are other important morphogens I would like to mention. One of them is Wnt. So Wnt binds to a receptor called frizzle, and then it sends a signal to this protein complex. What it does is degrade a certain protein called beta-catenin. And usually this complex degrades beta-catenin, but frizzle, when it gets signal, and then it inhibits this degradation. So beta-catenin is actually in this cadherin complex. So cadherin binds to beta-catenin, and then beta-catenin binds to alpha-catenin, and then binds to actin filament. So it's a part of cell adhesion component. So usually, like I said, neural crest has a lot of cadherin stick together. So it's in the beta-catenin is in the complex, but sometimes it comes out, especially when snail started to express, and then beta-catenin, cadherin is gone, and then beta-catenin will be released. Usually it gets degraded, but if you have Wnt signaling, so the main component that recognizes beta-catenin is called APC. APC recognizes beta-catenin because once it's released, it's phosphorylated by this kinase called GSK3 beta. But if you have Wnt signaling, what it does is it inhibits this GSK3 beta, so it's no longer phosphorylated. So it's not going to be degraded and accumulated in the cytosol. And once it's accumulated, it likes to go to nucleus and then bind its partner called LEP or TCF, and then this complex will bind DNA turn on the gene. So this is one of the very important morphogen. Another thing I would like to mention is this retinoic acid array. Retinoic acid is very hydrophobic, so it crosses membrane, plasma membrane. It goes to cytosol. And the receptor is sitting in the cytosol and binds directly in the cytosol. And it goes to a nucleus and turns on the gene. Is this familiar to you? We saw something like this the other day. What was that? Hormone, the stress hormone, glucocorticoid, which crosses the membrane, and the receptor is not on the membrane, it's in the cytosol, and then binds to it, and then goes to a nucleus, right? So, retinoic acid is created from this precursor material, which is retinol, or vitamin A. Vitamin A is abundant in carrots. Do you like carrots?
Sure.
But a lot of kids don't like them. And their mothers will say, oh, you have to eat it. It's good for your eye. you make a transgenic mouse, so it's manipulated in a way that it has this gene expression is regulated by retinoic acid, and then once it's transcribed, and then it turns blue. And this, if you look at this embryo of this type of transgenic mouse, this dark thing is where the retinoic acid is signaling, and sure enough, it's highly expressed in eyes. So your mom was right. So if you lack vitamin A, and then your vision will be impaired, and that's still a pretty serious problem in a developing country. So too little vitamin A is a serious problem. However, it turned out too much is also a problem. And we learned that in a hard way. So there was a drug called Accutane to treat with acne. And some pregnant women use that, and then it turned out that gives birth defect. So if you give vitamin A to a mother mouse, and then the embryo looks like this, so many ways you get retinoic acid, retinoic acid signaling going on, and the brain looks. This is the normal brain, so this is the retinoic acid treated brain, grossly deformed. So too little, bad, but too much is also bad. Some of the vitamins, so vitamins, usually you have to take more, but not all the vitamins you can take like 1,000 times more. So we've been looking at how to organize dorsal ventral axis, but what about anterior posterior axis? So let's think about that next. So brain will be formed in this way. So the most anterior part to the most posterior part, there are different parts created. The first one, the most anterior part, is called presencephalon, and then mesencephalon, lumbencephalon, and then the spinal cord. And then as you grow, so this part is becoming to bend, right? And then as you grow, this prosencephalon will become two parts, telencephalon and diencephalon. And lumbencephalon will become metencephalon and myelencephalon. And it's nicely color-coded. You can see what happens. And then those parts create different parts of the brain. So telencephalon will become basically cortex, which is the largest part in the human, but also hippocampus, basal ganglia, and the olfactory bulb. And then diencephalon will be kind of inside in our brain. Will become thalamus, hypothalamus, and then mesencephalon will become midbrain. And it follows to metencephalon, and this will become cerebellum and the pons, and then myelencephalon becomes brainstem, and then going to spinal cord. So that's how brain is formed. So there is an ordered structure, anterior and posterior. How is this regulated? Well, turned out this gene called HOX is regulating. And it was initially discovered in the Drosophila. So when you're looking at the Drosophila gene, they encounter this repeated DNA sequence. It comes again and again and again. And they call it homeobox. It turned out that encodes a part of DNA part of protein domain, which is actually DNA binding domain. So it's homeodomain. It turned out because they are encoding series of transcription factors, and it has this particular DNA binding domain, homeodomain. So these are called Hox proteins. And if you look at which Hox genes are expressed, as you can see, it's very nicely color-coded. It's sequentially expressed from anterior to posterior. And it turned out human is basically the same. It's not. We are not too different from Drosophila. We have the same plan, although we have a little more complicated gene system. We have 4 Hox genes, and each of them has sequential expression. But that's how you can organize different parts from anterior to posterior. Do you have any question? Because I have a question. It's like, but how do you know I'm really the most anterior, and so I will express this, or I'm close to the tail, so I will express this. How do you know that? It turned out there is a gradient. For example, bicoid. It has a strong gradient from anterior to posterior. Messenger RNA of bicoid actually comes from egg. So it already has this messenger RNA even before fertilization. So we call it maternal effect chain. What it does is 2 things. One thing bicoid does is to suppress codon messenger RNA translation. Another thing is induce anterior gene expression. So 2 things. And that way, you get this gradient, and as gradient changes, different genes will be expressed. So far we looked at positional effect, and we looked at dorsal ventral axis, which is organized by 2 morphogenes, sonic hedgehog and BMP. And another thing is anterior posterior axis, and Hox proteins organize that. So let's move on to timing issue. So if you look at cortex, so they did this experiment. They injected radioactive material, actually radioactive nucleotide, in a different time point of the development, and then look at where it ends up. And here's the cortex. Cortex has layers, usually 6 layers. And if you look at the radioactivity, the area injected ends up in a deep layer. And then if you inject the radioactive material later on, it ends up in a superficial layer. So you can tell this cortex was made inside out manner. So this different layer is formed by at a different timing. So how is it organized? So I mentioned about the neural stem cell or neural progenitor cells, and they keep generating, or most of the time they replicate itself, and then sometimes it generates neurons. And it happens in this between this base pile surface and the neural tube, which is called ventricular zone. And as neural progenitor cell goes through mitosis, interesting happens. Well, let's think about mitosis. So there is several steps when you do mitosis. First thing they do is synthesize DNA. So they double the DNA, right? That's called S phase, S stage. And then there's a gap, and then they go mitosis, divide. And if you look at where the cell body of neuroprogenitor cells, it goes up during this S stage, and then goes down again, and cell divides from here. And when they're going through self-replication, they divide this way. But when they differentiate to generate neuron, it goes this way. So they go up and down, and then either this or this. So how is this generating neuron signaling is done? Is done by a signaling molecule called Notch. So Notch has a ligand called delta. Interestingly, they are both membrane protein. So delta is not the secreted protein diffused. No, it's still in the membrane of some other cell. And then when they interact each other like that, it will induce cleavage of Notch intracellular domain. And this intracellular domain will go to nucleus and then bind and turn on His gene. So His gene has another typical DNA binding motif, which is called basic Heyx loop Heyx DNA binding motif, which looks like this. So this is another very often you encounter by DNA binding in the transcription factor. So if you hear that, you will know what it is. Another one we mentioned was zinc finger, right? So the problem is you want to generate different types of neuron as time go by. So you want to do it sequentially generating. You don't want to just make tons of neurons and then end. Then you can make this different layer. So you want to regulate the pace of how to generate neurons. And how is it done? It's done by this Notch and delta. Interesting thing is, so the middle cell will become a neuron. So you get more Notch signaling, and then Notch signaling will create gene expression which is required for neuron. But at the same time, this ligand delta, because it's a membrane protein, can send this signal to other cells, the other side. So it knows that the Notch was cleaved, and then that is sensed by this neighboring cell. And what happens is it turns on the Notch signaling in neighboring cell. So it will be harder for them to become neurons because you can't get Notch signaling. So if there is one neuron differentiating, neighboring cells will be suppressed to become differentiation, which is called lateral inhibition. And there is another lateral inhibition in neuroscience. Don't get confused. This is lateral inhibition during the development. So when they are generated, what happens next? Well, so this is the ventricle, and the pile surface is here, and they migrate from here to there. So that's shown magnified here. So there is a cell type called radioglia, and it's like a scaffold. It has radio structure that's radioglia provides. And then neuron was born on this side, not the pile side, this side, and then will migrate out. Here's an illustration of that. So this is the radioglia, and then green thing, young neuron, will migrate. But when they migrate to the top of this radioglia cell, here there is a signaling molecule called reading. What it does is to signal this migrating neuron, get off, stop migrating. So this is the migrating neuron in green, and then this red thing is the reading. Yes, so let's talk about this reading. So what happens is radioglia cell itself will grow, so it becomes bigger and bigger as you develop. In the meanwhile, neurons will migrate, but then if you hit reading, and then it comes off, stop. And then the new neurons are migrating until it hits reading. So these neurons are already stuck there, but then next neuron will keep migrating until it hits reading. That's how you make inside-out pattern of layer formation, right? What happens if you don't have reading? If you don't have reading, those neurons don't get any signal to say get out. So it keeps growing, clinging onto it. But in the meanwhile, the new neuron will be generated, so it gets stuck because the first neuron is still hanging there. As a result, you will get exactly opposite layer formation. It's a striking thing. So here's the nice image of neuron migrating along the radioglia. The radioglia soma is in the arrowhead, and migrating neurons are shown in the arrow. Like this one, I can see really nicely migrating. And when they were doing this experiment, from time to time, they saw something like this shown in the star. It's not really migrating. But they didn't see it often, just occasionally. They see it. It's like, what's that? They couldn't tell where those neurons come from. So they were thinking, maybe sometimes they just fall off and then go like, ah. Maybe. And it took a long time for them to figure out what those neurons were. It turned out those neurons are coming from very far away. It comes from ganglionic eminence. There are two parts, lateral and medial ganglionic eminence, so LGE and MGE. And those neurons are inhibitory neurons, interneurons. So they migrate all over the brain, including cortex, hippocampus, everywhere. So they migrate quite differently, and they migrate long distance. And that's what they were looking at. And if you look at cortex, inhibitory neurons are only 10% or 15% at most. So as you can imagine, They don't see it often. Only occasionally. Oh wait, what was that? That was the internet. So we were looking at timing issue. So these timing issue and the positional issue, and the interneurons follows this rule too. So there is LGE and MGE, different portion of LGE, ganglion neurons, produce different types of interneurons. And timing issue too. So as they develop, they start producing different types of interneurons. And there are so many different types of interneurons. So in addition to making different types of neurons, timing is important for producing other neuronal subtypes. For example, oligodendrocytes will be produced after they were producing neurons. So they produce neurons first, and then oligodendrocytes and astrocytes. So the timing governs the neuronal cell type, not only types of neurons. So let's just summarize what we learned today. There's two things important. One is position, another is timing. And regarding position, dorsal ventral axis is governed by two morphogens, sonic hedgehog and BMP. And anterior, posterior is organized by Hobbs proteins. And for timing, we learned that cortex layer will form sequentially in an inside-out manner. And we also mentioned that gliogenesis also regulated by the timing. Okay. Any questions? No questions? All right. Okay, then that's it.